Today we're going microscopic.
Yep.
And I don't mean just looking at a bug on a leaf kind of microscopic. I mean we are descending into a world that exists on a scale so small it totally defies intuition.
Right. It's an entirely different universe down there.
Yeah, and yet it dictates the course of human history, personal health, and sometimes life and death. For you listening right now, we were taking a massive stack of research, specifically a very dense, very detailed chapter on medical microbiology. Chapter 14 to be exact.
A classic chapter.
It really is. And our mission today is to turn that dense textbook material into an exploration of a civil war that is happening right now, quite possibly inside your own body.
It is a war, quite literally. It's this constant, unforgiving battle for territory, for resources, and basically for survival. And the combatants that we're profiling today are some of the most successful, most versatile, and frankly, most terrifying organisms on the planet. We are looking at the Streptococci and the Enterococci.
Now, I have to admit, before digging into the source material, the word strep to me just meant, a sore throat.
Sure. Most people think that.
You feel scratchy, you go to the urgent care, you get a quick swab, maybe they give you some amoxicillin and you watch Netflix for 2 days while you recover. But looking at this material, strep throat is, it's barely the tip of the iceberg.
Barely the tip. It's actually astounding how broad their impact is.
Right, because we're talking about flesh-eating disease, we're talking about fatal meningitis, we're talking about tooth decay, and we're talking about bacteria that are so unbelievably tough they can survive in bleach and salt.
Exactly. The range is what makes this family of bacteria so fascinating to study and also so incredibly difficult to treat in a clinical setting.
It's a paradox.
Total paradox. On one hand, you have organisms that are completely harmless residents of your mouth or your gut, your normal flora.
Yeah.
They're just paying rent, hanging out, and actually helping you in some ways by outcompeting worse bugs.
Yeah.
But on the other hand, you have organisms from the exact same family, sometimes totally indistinguishable under a simple microscope, that, if they get into the wrong place at the wrong time, can kill a healthy adult in 24 hours.
It's that Dr. Jekyll and Mr. Hyde nature of these things that really hooked me.
Yeah.
So for everyone listening, let's set the stage visually. If we were to shrink down, magic school bus style and look at these things under a high-powered microscope, what are we actually seeing?
You'd be looking at spheres. That's what the word cocci means in microbiology, berries or spheres.
Okay, spheres.
Right. And these are gram-positive cocci, which means they have a very thick cell wall that holds onto a purple dye when we stain them in the lab. But the real signature look for Streptococcus, and what separates it from something like staph, is how they arrange themselves.
Because staph looks different.
Yeah, staph likes to bunch up in these clusters, kind of like grapes. Strep is, it's more elegant. They form pairs or long twisting chains. Think of a purple pearl necklace floating in fluid.
A pearl necklace? That sounds so innocent.
It does, doesn't it? It looks genuinely beautiful under the microscope. Purple spheres in these long, twisting chains. But as we're about to find out, those pearls can be incredibly deadly.
Yeah, no kidding.
And historically, the first major challenge scientists faced was just figuring out who was who. You look under the scope, they all look like pearls. So how do you tell the harmless mouth bacteria from the flesh-eating monster? Just by looking at the shape.
You can't. Which brings us to what the notes call the sorting hat of microbiology, the blood agar test. And this fascinated me because it feels so, medieval. You basically feed them blood to see how they react.
It is a bit primal, I'll give you that.
Yeah.
But it's still the gold standard for a quick classification in any clinical lab today. It's called hemolysis.
Hemolysis, literally blood splitting.
Exactly. You grow the bacteria on a petri dish filled with nutrient jelly that's been mixed with actual sheep blood. Yeah. And then you just wait to see what the bacteria do to the red blood cells around them. It's a rough way to categorize them, but it neatly divides the family into 3 distinct clans.
Okay, so let's break down these clans for the listener. We have alpha, beta, and gamma. Based on the chapter, beta seems to be the one you really, really don't want to mess with.
Generally, yes. The beta hemolytic Streptococci are the highly aggressive ones. Beta means complete hemolysis.
Complete destruction.
Total destruction. If you hold that petri dish up to the light, you will see a clear, completely transparent halo around the bacterial colonies.
Because they've basically exploded the blood cells.
They've obliterated them. They release specific toxins that rupture the red blood cells and fully digest the hemoglobin inside. It's a scorched earth policy. They clear the area entirely so they can scavenge the iron and nutrients.
Yikes. Okay, so beta is the clear zone, the scorched earth. What about alpha?
Alpha is more of a partial destruction. They damage the red blood cells and leak out the potassium and the hemoglobin, but they don't wipe them out completely.
So what does that look like on the plate?
Well, chemically, the hemoglobin gets oxidized by hydrogen peroxide that the bacteria produce. This oxidization turns the hemoglobin green. So on the red plate, you see this greenish, somewhat bruised appearance right around the colony.
Greenish. Okay, that rings a bell. That sounds like the viridins group the notes talk about later.
You nailed it. Viridis is Latin for green. So the viridins strep are the green-producing alpha hemolytic ones. And then finally, you have gamma.
The pacifists.
Right, the pacifists. Gamma hemolysis is actually a misnomer because it means a total lack of hemolysis. They grow on the blood, they use the basic nutrients in the agar jelly, but they don't harm the red blood cells at all. The agar stays perfectly red right up to the very edge of the colony.
Okay, so beta is the one we're really worried about initially because of that clear zone of destruction. But the notes mention that just seeing a clear zone on a plate wasn't specific enough for doctors. Like, it's a start, but there was a scientist, Rebecca Lansfield, who realized we needed a much better filing system.
Oh, Rebecca Lansfield is an absolute legend in microbiology. Back in the 1930s, she realized that just looking at blood destruction was way too broad. You had different diseases causing beta hemolysis. So she developed a serologic classification system.
Serologic meaning based on antibodies.
Yes. Basically, she looked at the carbohydrates, the very specific sugars that stick out of the bacteria's cell wall.
Like a molecular ID card.
Precisely like an ID card. Or a jersey number on a football player. She categorized them into Group A, Group B, Group C, and so on, all the way down the alphabet. It's based purely on that specific sugar antigen on their surface.
That makes sense.
So when doctors today talk about having Group A strep, they are directly using Lansfield's system from almost a century ago. It was revolutionary because it finally allowed us to track outbreaks accurately and understand that specific groups caused very specific diseases.
Now, of course, modern genetics has come in recently and complicated everything by sequencing the DNA, right? Proving that some of these phenotypic distant cousins are actually genetic siblings.
Oh, DNA sequencing ruined the neat little boxes, for sure.
But for the sake of understanding the diseases and treating patients, the medical community still really sticks to the classics, hemolysis and Lansfield groups.
Without a doubt, that's still the most practical way to think about it clinically. When a sick patient comes into the ER, you don't necessarily need their full genome in minute 1. You want to know, is it Group A? Is it Group B? How aggressive is it?
Right. So let's talk about the big one. Public enemy number 1, the source material calls this the prototypical human pathogen. Group A, beta hemolytic Streptococcus, also known scientifically as Streptococcus pyogenes.
Streptococcus pyogenes, and honestly, the species name tells you everything you need to know. Pyogenes literally translates to pus-making.
Pus-making. Lovely. Just what you want to hear. So this is the bug behind strep throat, but it's also the bug behind scarlet fever and the flesh-eating disease.
Yes, all of them.
It just seems insane to me that one single type of bacteria can cause a slightly scratchy throat in a kid and also completely dissolve a grown man's leg. How does it manage to be so versatile? It seems like it has a toolkit full of highly specialized weapons.
It has an arsenal that would make a military general jealous. And to truly understand why it's so dangerous, we have to look closely at its anatomy. Let's start with its disguise. This bacteria wears a literal camouflage cloak made of hyaluronic acid.
Wait, hyaluronic acid. I see that in commercials for expensive skin creams all the time. It's supposed to be moisturizing, right? Plumping the skin.
It is. But more importantly for our story, it's a completely natural component of your own connective tissue. It's the molecular goo that holds your human cells together. It is everywhere in your body.
Okay.
So Streptococcus pyogenes evolved to cover itself in a thick outer capsule made of this exact same human stuff.
That is deeply devious. So the immune system is patrolling the bloodstream, sees the hyaluronic acid capsule and goes, oh, nothing to see here, that's just us.
Exactly. It's a perfect example of molecular mimicry. This capsule prevents phagocytosis.
Phagocytosis being when your immune cells eat the invaders.
Right. Macrophages and neutrophils are your pac-men. Usually a macrophage grabs a bacteria because its surface feels foreign, but this group A strep feels like self. The macrophage literally glides right off its slippery surface. It is an invisibility cloak.
That is terrifying brilliance. Okay, so it's invisible to the early responders, but it's not just hiding, obviously, it's attacking. The text mentions something else on the surface called the M protein. and it describes it as spiky hair or fuzz on the surface of the bacteria.
The M protein is arguably the most important virulence factor this bacteria possesses. Visually, yes, under an electron microscope it looks like fuzz, but functionally it's a deflector shield and a disruptor. It extends way out from the cell wall and physically interferes with the human complement system.
The complement system, for the listener, that's the part of the immune system that tags bacteria for destruction, right? Like painting a target on them.
Exactly. It's a cascade of proteins in your blood that coat bacteria to broadcast an eat me signal to the immune cells. The M protein binds to a specific regulator in your blood called factor H and basically hijacks it to turn off that eat me signal.
Unbelievable.
But here's the real kicker, and this answers a question a lot of parents have. This is the reason why you don't just get strep throat once in kindergarten and then never again.
Right. Why do we keep getting it?
Because there isn't just one M protein. There are over 150 different types.
150 types. So if I get immunity to type 1.
You are immune to type 1 forever. Your body remembers that specific fuzz, but you are totally vulnerable to type 12 or type 80. This is why you can get strep throat multiple times as a kid. You're building immunity to specific M proteins one by one, but the bacteria has this massive wardrobe of disguises to cycle through.
That feels incredibly unfair. Okay, so it has disguise, it has a deflector shield, it has 150 outfit changes. And let's talk about the heavy artillery, the toxins and the enzymes it pumps out. Because reading this list in the chapter, it reads like a chemical warfare manifest.
It really does. Let's look at streptokinase first. This is an enzyme that dissolves blood clots. Now, mechanically speaking, ask yourself, why would a bacteria want to dissolve a blood clot?
Well, usually when you get an infection or a cut, your body clots blood to stop bleeding, but also to trap bacteria, right? Like putting up a barricade to quarantine the area.
Exactly right. The body tries to wall off the infection with fibrin clots. It's a containment strategy. Streptokinase is the bacteria's genius counter-adaptation. It actually transforms your own plasminogen, which is an inactive protein floating in your blood, into plasmin.
And plasmin does what?
Plasmin is essentially a molecular pair of scissors that cuts fibrin.
So the bacteria hijacks your own circulating protein to cut the wires of the fence your body just built around it.
It's a jailbreak enzyme. It dissolves the prison wall so the bacteria can escape the local area and spread rapidly through the tissue. And, ironically, medical science harvested this weapon. For years, doctors used purified streptokinase as a frontline drug to treat heart attacks and strokes.
Wait, really?
Yeah, to bust the clots that were blocking human arteries. We were literally using the bacteria's own escape weapon to solve our cardiovascular problems.
That is such a cool twist. Wow. Okay, what about DNase? This is the next enzyme on the list. And I have to say, this one sounds particularly gross.
It is gross, but it's fascinating from a fluid dynamics perspective. Think about pus. When you have a bacterial infection, your immune cells, specifically neutrophils, rush to the site. They fight, they consume bacteria, and they die in massive numbers.
Right.
When they die, they explode and release their own DNA into the environment. Now, DNA is an incredibly sticky, viscous molecule. It's basically molecular glue. That is actually what makes pus thick and stringy.
So the battlefield gets clogged up with this sticky DNA goo from the dead immune soldiers.
Yes. And that sticky goo physically traps the bacteria. It slows their movement to a crawl. So Streptococcus pyogenes releases DNase, an enzyme that specifically chops up long strands of DNA. It literally liquefies the pus.
It turns the battlefield from a sticky swamp into a slip and slide.
Precisely. It reduces the viscosity of the fluid, allowing the infection to spread freely through tissue planes.
Unbelievable.
And then on top of that, you have hyaluronidase. We call it the spreading factor. Remember how the bacteria's capsule is made of hyaluronic acid?
Yeah, the human tissue cement.
Well, the bacteria also makes an enzyme that digests hyaluronic acid.
Wait a minute, won't that dissolve its own invisibility cloak?
It can, but the bacteria controls the timing. The primary target is your tissue. Hyaluronic acid is the mortar holding your cells together. This enzyme dissolves that mortar. It literally opens physical gaps between your cells so the bacteria can march deeper and deeper into the body.
So it wears the cement as a coat to hide, but carries a jackhammer to break your cement when it wants to move.
You're getting the picture. This is an aggressively invasive organism. It doesn't want to just sit politely on the surface of your throat. It actively wants to dive deep into the tissue.
And we haven't even talked about the superantigens yet, the pyrogenic exotoxins. These are the ones behind toxic shock syndrome, right? Because the mechanism here seems completely different from just dissolving tissue locally.
It is completely different. This is psychological warfare on a cellular level. These are the SPAD toxins, streptococcal pyrogenic exotoxins, usually types A, B, and C. We classify them as superantigens.
Superantigens, how do they work?
Normally, your immune system is highly disciplined. A T cell, which is like the general of the immune army, only activates when it sees a very specific enemy presented to it by an antigen-presenting cell. It's a molecular lock-and-key handshake. Extremely specific.
Like a secure, classified military briefing. You only get the info if you have the exact right clearance.
Right. Because of that specificity, only about 0.01% of your T cells usually respond to any given infection, just the specialists. But a superantigen cheats the system. It forces the handshake. It binds to the outside of the T cell and the antigen-presenting cell and forcibly locks them together, regardless of whether they actually match.
It hacks the communication lines. It bypasses the security check entirely.
Exactly. And because it's nonspecific, it activates up to 20% of your entire T cell population all at once.
20%.
Yes. That is millions upon millions of cells releasing attack signals called cytokines simultaneously into your bloodstream.
And that's what we call a cytokine storm.
Exactly. The body is completely flooded with inflammatory signals. Blood vessels dilate massively and start leaking fluid. Your blood pressure crashes and your major organs start failing from lack of perfusion. It's not the bacteria directly killing the organs. It's the confused, overreacting immune system burning its own house down to catch a single burglar.
Incredible.
Sure.
So with all these weapons, the clot buster, the pus liquefier, the super antigens, let's transition to what happens when this thing actually infects you. Segment 2 in our deep dive. Clinical chaos.
The clinical presentation of Group A strep depends entirely on how deep the invasion goes. If it stays right on the surface, on the mucosa, you get pharyngitis, strep throat, or on the skin, you get impetigo, those crusty, honey-colored sores you often see around kids' mouths.
Very common.
But if it uses those enzymes to go deeper.
Then we get into the scary stuff. Erysipelas. I looked up clinical photos of this while reading the source text. It looks like a bright red map drawn with a marker on someone's face or leg.
It's very distinctive visually. Erysipelas involves the upper dermis and the superficial lymphatics. The key diagnostic feature is the edge of the rash. It's raised, it's bright red, and it has a very sharp, clear border. You can literally run your finger over it and feel the step off from normal skin to infected skin, and it spreads rapidly.
And if the bacteria pushes deeper than the upper dermis,
then you get cellulitis. That's an infection in the deeper subcutaneous tissue. The borders there are less defined, the skin is pinker, much more swollen and warm. But the absolute nightmare scenario, the one that makes every ER doctor sweat, is necrotizing fasciitis.
the flesh-eating disease.
That's the tabloid name, sure, but necrotizing fasciitis describes exactly what it is medically. Necrotizing means dying or dead tissue. Fasciitis means inflammation of the fascia. The fascia is that tough white sheet of fibrous connective tissue that wraps around your muscles and organs.
So the bacteria gets all the way down into that deep layer.
Yes. And remember the hyaluronidase, the spreading factor. It uses that to race along the fascial plane. It moves horizontally, deep underneath the skin. You might look at the skin surface and just see a small red mark or a blister, but underneath, in the fascia, the infection has already spread 6 inches up the patient's arm.
That is the scary part.
Yeah.
What you see on the surface isn't what you get. The damage is hidden.
And as it spreads along the fascia, it destroys the blood vessels that feed the skin and the muscle above it. The tissue literally turns black and dies from lack of blood. This infection can move at a rate of inches per hour. A surgeon can literally mark the edge of the redness with a pen, come back in 30 minutes, and the redness has jumped past the line.
So in a case like that, won't IV antibiotics just kill it?
They can't reach the infection. That's the tragic part. The local blood supply is dead. The antibiotics are floating in the patient's bloodstream, but the biological road to the infection site is washed out.
Oh, wow.
The only treatment that works is rapid surgical debridement. You have to take the patient to the OR, cut them open, and physically remove every single piece of dead and infected tissue. You have to keep cutting until the tissue bleeds, because bleeding means it has a blood supply, which means it's alive and the antibiotics can reach it.
It's horrifying to think that this is the exact same bug that causes a mild sore throat in a 7-year-old.
Same bug, same genome, just a different location in the body and different virulence genes turned on. And let's not forget puerperal fever, also known as childbed fever. Historically, before modern medicine, this was a massive killer of young women.
Right. Because doctors would go from doing autopsies to delivering babies without washing their hands.
Yes, they didn't understand germ theory. They were mechanically delivering group A strep directly into the highly vascular, vulnerable uterus of a woman who just gave birth.
That was Ignaz Semmelweis's huge discovery, right?
Exactly. He realized that simply having doctors wash their hands with a chlorine solution lowered the maternal mortality rate in his ward from 18% down to about 1%. That invisible killer he was washing away was almost certainly group A strep.
Astonishing. Now, we also have the toxin-mediated diseases in this section. Scarlet fever. Honestly, this sounds like something out of a Victorian novel, like Little Women or something, but the text says it's still very much around.
It is definitely still around. It's basically strep throat, but with a systemic complication. The bacteria localized in the throat releases that pyrogenic exotoxin we mentioned earlier, usually SPIA or SPEC. The toxin enters the bloodstream and spreads throughout the entire body. It causes a diffuse, bright red rash that feels rough to the touch, like fine sandpaper.
And it does something weird to the tongue too, right?
The classic strawberry tongue. At first, the tongue has this thick white coating with swollen red papillae poking through. That's the white strawberry stage. Then after a few days, the white coating peels off completely, and the tongue is beefy red with prominent bumps. The red strawberry.
It's a classic board exam question for med students.
So that's the acute phase. But the chapter emphasizes that there is a much darker side to group A strep that isn't about the acute infection at all.
Sure.
It's about segment 3, the aftershocks. These are diseases that happen weeks after the initial infection is completely gone.
This part confused me a bit at first. So the bacteria is completely dead. You took your penicillin or your immune system won the war. You feel fine. But then weeks later, you get seriously sick again.
Exactly. These are called post-streptococcal sequelae. It's crucial to understand that it's not an active infection anymore. It's an autoimmune disaster. Your body is attacking itself. The two big ones we worry about are acute glomerulonephritis, or AGN, and rheumatic fever.
Let's start with the kidneys, AGN. How does a throat or skin bug ruin your kidneys weeks later?
This is what we call a type 3 hypersensitivity reaction. Basically, during the active strep infection, your body made massive amounts of antibodies to fight the bacteria. These antibodies bind to leftover, dead pieces of the bacteria floating in your blood, forming what we call immune complexes. They are microscopic clumps of antibody and antigen stuck together.
And these sticky clumps have to go somewhere to be cleared out.
They travel through the bloodstream to the kidneys. The kidneys are your body's filters. These complexes get physically jammed in the tiny microscopic filters of the kidney, which are called the glomeruli.
Like clogging a drain with hair.
Very similar idea, but it's an inflammatory clog. The trapped complexes activate the complement system right there inside the kidney filter. The kidneys become severely inflamed, and they can't filter blood properly anymore. You retain fluid, you get high blood pressure, swelling in the face, especially around the eyes, and the classic textbook sign, cola-colored or coffee-colored urine.
Which is actually old, oxidized blood leaking through the damaged filter.
Right. Now, AGN usually happens after strep skin infections, like Impetigo. But the other major aftershock, rheumatic fever, classically follows throat infections. And this is the true historical tragedy of untreated strep throat.
The mechanism here is that molecular mimicry concept again. This goes all the way back to that M protein fuzz we talked about at the beginning, right?
It does. Remember we said the bacteria wears a disguise to evade detection? Well, sometimes a disguise is a little too good. Certain types of M protein look incredibly similar structurally to proteins found naturally on your own human heart valves, specifically a protein called cardiac myosin and in your joint tissues.
So the antibodies you purposefully made to kill the strep.
They get confused. The infection is completely over, the bacteria are dead and gone, but those highly specific antibodies are still patrolling your blood. They bump into the mitral valve in your heart, look at the myosin, and say, hey, that looks exactly like the M protein. And they signal an attack. They attack the joints, causing a severe migrating arthritis, and they attack the heart tissue, causing acute rheumatic fever.
The old medical phrase in the text was, it licks the joints but bites the heart.
That's the old adage, and it's perfectly accurate. The joint pain is excruciating. It moves from knee to ankle to wrist, but it eventually goes away permanently without leaving any lasting damage. The heart valve damage, however, creates permanent scars. The delicate leaflets of the valves get thickened, fused, and stiff. This leads to severe rheumatic heart disease, which can cause total heart failure 10, 20, or even 30 years later.
And this right here is why we treat strep throat so aggressively, isn't it? I always honestly thought antibiotics were just to make my throat stop hurting a day or two faster.
Not at all. Strep throat will almost always resolve entirely on its own in a few days. The pain will go away. We give antibiotics primarily to kill the bacteria quickly before the immune system has time to ramp up that massive, confused antibody response that causes rheumatic fever. We are treating the throat today to save the heart tomorrow.
That is such a crucial point for people to understand. Okay, let's pivot. We spent a ton of time on Group A because it's obviously such a major villain in human history. But moving down the Lancefield alphabet, we hit Group B, Streptococcus agalactiae.
Group B strep. Now, this organism operates on a completely different biological philosophy. Group A is an aggressive invader. Group B is a quiet colonizer. For most adults, and specifically for about 25% of healthy women, it is just normal flora. It lives happily in the lower GI tract and the vaginal canal. It causes absolutely zero symptoms.
Totally harmless to the adult carrier.
Usually, yes, it's just hanging out. But the critical danger arises during childbirth. If a baby is born vaginally through a birth canal that is colonized with Group B strep, the baby is exposed to a massive load of these bacteria. And newborns have incredibly immature immune systems. They don't have the specific complement proteins or the maternal antibodies needed to fight off this particular bug.
So the mother is perfectly fine, but the baby is in sudden mortal danger.
Correct. Group B strep can cause devastating neonatal sepsis, pneumonia, and meningitis in the first few days or weeks of life. Before we had a strategy for this, it was the #1 killer of newborns due to infection in the developed world.
So how do we stop it? What's the protocol now?
We screen. Preventative medicine. Between 35 and 37 weeks of pregnancy, every expectant mother gets a routine swab. If she tests positive for carrying Group B strep, we actually don't treat her right then.
Why not? Just wipe it out early.
Because it's part of her normal flora. If you give her antibiotics at 35 weeks, you'll kill it, but the bacteria from the GI tract will just recolonize the area within a week. It would just come back before delivery. So we wait. We wait until labor actually starts.
And then what's the move?
As soon as her water breaks or active labor begins, we start her on high-dose IV penicillin. The goal isn't to cure the mother permanently. The goal is just to knock down the bacterial count in the birth canal to 0 for those few critical hours while the baby is physically passing through. It's basically like chemically sanitizing a hallway right before a very vulnerable VIP walks down it.
That strategy has been a massive public health success, hasn't it?
Huge. Neonatal Group B strep cases have plummeted since we instituted universal screening and intrapartum antibiotics. However, microbes always adapt. We are now seeing a strange new trend. Group B strep is starting to cause very serious skin, bone, and bloodstream infections in elderly adults and people with diabetes. It's an opportunist that's finding a new immunologically vulnerable population to exploit.
Evolution literally never stops.
Okay, let's move quickly through the other Lancefield groups before we hit the viridins, groups C and G.
They are the lookalikes. On a blood agar petri dish, they look exactly like Group A large colonies, strong beta-hemolytic clear zones. And they can definitely cause bad sore throats too. But clinically, the major difference is that they rarely, if ever, trigger rheumatic fever. They're like Group A's slightly less ambitious cousins. You treat them, but you don't panic about the heart.
Good to know. And Group D. Now this one had a real Sherlock Holmes deductive moment in the text that I loved. Streptococcus galolyticus, which used to be known as Streptococcus bovis.
Ah, yes, the S. bovis group. These are usually gamma or alpha hemolytic, not very aggressive on the plate. But here's the classic clinical pearl that literally saves lives. If a doctor draws blood from a patient with a fever and the lab finds S. galolyticus growing in that blood culture, meaning the bacteria has somehow breached the gut and gotten into the systemic bloodstream, the very next step, absolutely mandatory, is to schedule a colonoscopy. Why? What is the biological connection between a random blood infection and checking the colon?
There is an incredibly strong documented statistical association between this specific bacteria and the presence of colon cancer or precancerous polyps. We believe the bacteria fundamentally loves the specific microenvironment of a colon tumor. Maybe it's the specific nutrients the tumor leaks, maybe it's the altered architecture or the lack of oxygen, but it thrives and multiplies right on the surface of the tumor. And because tumors have very messy, leaky blood vessels, the bacteria easily spills over into the bloodstream.
So the bacteria is acting as a whistleblower. It's screaming, hey, I'm only in the blood because there's a tumor down here, check the colon.
Exactly. Finding the bacteria in the blood is very often the very first sign of a completely silent colon cancer. It gives oncologists a chance to catch it early before it metastasizes. It's a brilliant diagnostic clue.
I love how interconnected all of this is. And then there's the anginosis group. I have to mention them just because of the sensory detail in the book.
Streptococcus anginosus. These are part of the normal flora too, usually in the mouth or gut, but their main pathogenic trait is that they are profound abscess formers. If they get into deep tissues like the liver or the brain, they don't just spread out. They form massive walled-off pockets of thick pus. And yes, to your point, in the microbiology lab, when you grow certain species of this group, they produce a very distinct, volatile odor that smells strongly of butterscotch or caramel.
I feel like if your brain abscess smells like delicious caramel, you have much bigger problems to worry about.
Indeed. It's a very sweet smell for a very sour, life-threatening clinical situation.
All right, let's leave the pyogenic or pus-producing groups behind and move to segment 6, the viridins group. We established earlier that viridins means green, from the alpha hemolysis.
Correct. They bruise the blood, they don't clear it. And these are the absolute most common, prevalent residents of your mouth. For everyone listening, if you run your tongue over your teeth right now, you are physically licking millions of viridins streptococci.
Thanks for that vivid image. But mostly, these are the good guys, right? They keep worse stuff out.
Mostly, yes. They occupy the real estate so worse pathogens can't move in. Unless, of course, you feed them a lot of refined sugar. Let's talk about Streptococcus mutans. This particular viridin species is every dentist's absolute nemesis.
How exactly does it cause cavities? I know from childhood that sugar is bad for your teeth, but I never really understood the actual biochemical mechanism until reading this.
It's pure biochemistry. S. mutans possesses a specialized enzyme called glucosyltransferase. When you eat something sweet, it takes sucrose, regular table sugar, and breaks the molecule in half. It takes the glucose half and uses it to build these long, incredibly sticky, insoluble polymer chains called dextrans or glucans.
Dextrans, so it's synthesizing its own glue out of your candy bar.
It's building a fortress. That is exactly what dental plaque is. It's a thick, sticky biofilm glued directly to the enamel of your tooth. Inside that protective, sticky layer, millions of bacteria are safely housed. Then, they take the other half of the sugar molecule, the fructose, and they ferment it for energy. The byproduct of that fermentation is lactic acid.
And because of the dextrin glue, that lactic acid is trapped against the tooth.
Exactly the problem. If the bacteria were just floating freely in your saliva, the acid they make would just wash away or get neutralized by the buffers in your spit. But the plaque biofilm holds the acid directly against the tooth enamel. It acts like a concentrated acid compress, slowly leaching the calcium out and dissolving the hard enamel. That erosion is a cavity.
So the sugar feeds the bacteria, and the bacteria uses the sugar to build a sticky trap to hold acid against your tooth. That is so elegantly destructive.
Precisely. But the viridins group has another, far deadlier trick up its sleeve: subacute bacterial endocarditis.
This is the heart valve infection again, but this is distinct from rheumatic fever, right?
Very distinct, and a very important distinction to make. Rheumatic fever is an autoimmune mistake. The bacteria aren't actually in the heart, but endocarditis is an active physical infection. The bacteria themselves are physically growing on the leaflets of the heart valve.
So how do they get all the way from the plaque on your teeth down to the heart?
A simple, everyday break in the mucous membrane. A tiny cut in the gums from vigorous flossing, chewing something sharp, or, most commonly, invasive dental work, like a root canal or an extraction. The bacteria enter the bloodstream directly. We call this transient bacteremia. Usually, your immune system clears them instantly. They're gone in minutes. But if you happen to have a pre-existing damaged heart valve...
Damaged how?
Maybe from a congenital defect you were born with, or, ironically, from scarring caused by a previous bout of rheumatic fever. A healthy valve is perfectly smooth, like Teflon. Bacteria just slide right off. But a damaged valve is rough, scared, and creates turbulent blood flow.
And the bacteria snag on that rough patch.
Think of a fast-moving river. Debris flows right past smooth rocks. But if there's a jagged branch sticking out, things get snagged and accumulate. The bacteria settle on the microscopic rough spots of the damaged valve. They adhere, and they start to grow. They trigger the body to deposit platelets and fibrin over them, hiding them from the immune system, forming these large, cauliflower-like masses called vegetation. vegetations.
Vegetations.
Yeah.
That is an ominous medical term.
It's a literal mass of living bacteria and clotted blood flapping around inside the heart. Because the viridin strep aren't as wildly aggressive as staph or group A strep, they don't destroy the valve instantly. That's why we call it subacute. It happens slowly, insidiously, over weeks or even months. The patient just feels off. Low-grade fevers, chronic fatigue, unexplained weight loss, night sweats.
Silent destroyer.
It truly is, and it's notoriously difficult to treat because the bacteria are buried deep inside those vegetations, totally protected from circulating immune cells and somewhat shielded from antibiotics. It almost always requires 6 solid weeks of high-dose IV antibiotics and very often open-heart surgery to cut out and replace the destroyed valve.
Okay, moving from the mouth down into the lungs. Segment 7, we have to talk about the pneumococcus. Streptococcus pneumonia. This bug feels like a heavyweight champion of disease in this chapter.
Oh, it is. Sir William Osler, the famous physician, famously called it the captain of the men of death. Even today, with all our modern medicine, it remains a massive killer globally.
Visually, these look a bit different under the scope, too. The notes call them lancet-shaped diplococci.
Right. Instead of those long, twisting pearl necklaces, they usually hang out in pairs diplococci. And they aren't perfectly round, they are slightly pointed at the ends, like the sharp tip of a spear or a medical lancet. But the defining feature, the singular piece of armor that makes them a global killer, is the capsule.
We're back to the capsule.
Yes, but this one is the main event. It's a massive polysaccharide or complex sugar capsule. Without it, the pneumococcus is totally harmless. If you strip the capsule off in a lab and inject the bacteria into a mouse, the mouse's immune system cleans it up instantly without breaking a sweat. With the capsule, it is incredibly slippery and fiercely antiphagocytic. Macrophages just can't grab it. And the problem is, there are over 90 different serotypes of this capsule, 90 different flavors to fight.
And there's a really cool old-school lab test for this, too, right? The Quellung reaction, that's a German word, I think. It means swelling.
Yes, swelling. It's an elegant diagnostic test. If you take a sample of the bacteria and mix it on a slide with specific antibodies that perfectly match its capsule type, the antibodies bind to the capsule and cause it to absorb fluid and swell up massively under the microscope. It looks like the bacteria suddenly grew a huge, highly visible halo. It confirms exactly what type of pneumococcus you're dealing with.
So this bug, as deadly as it just hangs out in our nose and throat as normal flora, too.
Exactly. Depending on the season and the population, 40 to 70% of completely healthy humans are carriers. You or I might be carrying it right now deep in our nasopharynx.
So the obvious question is, if I'm carrying the captain of the men of death in my nose, why aren't I dead of pneumonia right now?
Because your mechanical and immunological respiratory defenses are excellent. The microscopic hairs called cilia beat upwards to form a mucus elevator, sweeping debris out. You have a strong cough reflex. You have alveolar macrophages patrolling the air sacs. Pneumococcus is, at its core, an opportunist. It waits. It strikes when those defenses are temporarily down.
Like when you catch the flu.
Exactly. That is the classic scenario. A primary viral infection, like influenza or COVID, strips away the protective ciliated lining of the airway. It literally paves a smooth road down into the lungs. That's why, historically, so many people who die during flu pandemics actually die of secondary bacterial pneumonia caused by pneumococcus, not the virus itself. Or think about severe alcohol intoxication. It suppresses the gag reflex, suppresses the cough, and even stuns the white blood cells. That opens the door wide for aspiration and infection.
And when it finally strikes and takes hold in the lungs, it causes classic pneumonia. The chapter describes the classic sign as rusty sputum.
That's the hallmark symptom. A patient experiences a sudden, brutal onset of shaking chills, a spiking high fever, sharp, stabbing chest pain when they breathe that's pleurisy, and a deep cough producing thick, rusty-colored mucus.
And what makes it rusty?
That color comes from red blood cells mixed with millions of dead inflammatory cells leaking from the capillaries directly into the alveoli, the tiny air sacs. The air sacs literally fill with inflammatory fluid, and without treatment, the patient essentially drowns in their own secretions.
But the invasion doesn't always stop at the lungs.
No, it does not. From the inflamed lungs, it can easily enter the bloodstream, causing bacteremia, and from there, it can cross the blood-brain barrier to cause fulminant meningitis. It is one of the leading causes of severe bacterial meningitis in adults. And at the other end of the spectrum, in toddlers and young kids, it's the absolute #1 cause of otitis media, extremely painful middle ear infections.
That blew my mind. So the routine ear infections my kid gets constantly and the lubber pneumonia my grandfather might get in a nursing home, it's the exact same pathogen.
The exact same bug, just different ages, different anatomy, different manifestations.
Let's talk about the vaccines for a minute, because this represents a truly fascinating bit of medical science history. We currently use two different types of vaccines for this, PCV13 and PPSV23. Why on earth do we need two completely different formulations for the same bacteria?
It solves a fundamental flaw in the human infant immune system. Remember we said the capsule, the bacteria's main weapon, is made of polysaccharide, just complex sugars.
Right.
An adult immune system can look at a massive, complex sugar molecule and say, that's foreign, I will make antibodies against it. That is exactly what the PPSV23 vaccine is. It's essentially a vial of purified capsule sugars harvested from the 23 most deadly types of pneumococcus. You inject it into an adult, and they mount a great defense.
That doesn't work for babies.
It does not work at all. The infant immune system, roughly under 2 years of age, is what we call T-cell independent for certain antigens, meaning it simply cannot see or process pure sugar antigens effectively. It essentially ignores them. So for decades, we had a vaccine that worked for adults, but we couldn't protect the babies who were dying of meningitis.
So how did the scientists fix it?
They developed the conjugate vaccine, which is the PCV13. They used an incredibly clever biochemical trick. They took the pure sugar from the bacterial capsule and they chemically they conjugated it to a highly immunogenic carrier protein, often a harmless piece of diphtheria toxin protein.
Why attach a protein to it?
Because while the baby's immune system ignores pure sugar, it absolutely loved recognizing foreign proteins. It is highly evolved to attack them. So the infant's immune cell grabs onto the carrier protein, pulls the whole complex inside to destroy it, and in the process, it gets forcibly exposed to the attached bacterial sugar. It tricks the immune system into processing the sugar via the much stronger T-cell dependent pathway.
It's a microscopic Trojan horse. Or like hiding medicine in a piece of cheese for a dog.
That is the perfect analogy. It completely bypasses the infant's blind spot by tagging the invisible sugar with a highly visible protein flag. And it has been a miracle of modern medicine. It has drastically reduced invasive pneumococcal disease and fatal meningitis in children worldwide since it was introduced.
That is just incredibly smart science. We have to move on to the absolute survivors of this story, the tough guys of the bacterial world. Segment 8. Enterococci.
Enterococcus faecalis and Enterococcus faecium. As the name entero implies, they are native residents of the enteric system, the human gut.
Now, historically, these guys used to be classified as Streptococci, right? Group D.
They were. For a long time, they were just another strep. But in the 1980s, deep genetic analysis proved they were distinct enough and, frankly, functionally different enough to get their very own genus. And honestly, they deserve their own category because they're absolute microscopic tanks.
How tough are we actually talking here?
We're talking extremes. Imagine a bacteria that can happily grow and divide in a 6.5% sodium chloride salt solution. Most normal bacteria would instantly shrivel up and die from osmotic shock. Imagine a bacteria that can easily survive in 40% bile salts. Bile is essentially harsh digestive detergent designed to destroy cell membranes. An Enterococcus swims in it. They can survive temperature fluctuations from a chilly 10 degrees Celsius all the way up to 45 degrees. And crucially for healthcare, they can live on dry, totally unhospitable surfaces like hospital bed rails, doorknobs, and stethoscopes for days or even weeks.
So they're incredibly hard to kill with standard cleaning. And that is what makes them such a massive problem in hospitals specifically.
They are the ultimate nosocomial, meaning hospital-acquired pathogens. Out in the real world, they don't usually hurt healthy people with intact immune systems, but put them in a modern hospital setting where patients are severely debilitated, where they have plastic IV lines and urinary catheters breaching their natural barriers, and most importantly, where patients are pumped full of broad-spectrum antibiotics that kill off all the competing friendly bacteria. Enterococcus just thrives. It takes over. It causes massive urinary tract infections, deep surgical wound infections, and notoriously difficult to treat endocarditis.
And the scary part, the part that keeps infectious disease doctors awake at night, is the resistance. The text says they just laugh at cephalosporin antibiotics.
They are intrinsically naturally resistant to a huge array of drugs we normally rely on. Cephalosporins simply don't bind to them, but the real existential fear is VRE. vancomycin-resistant Enterococci. Vancomycin is traditionally our big gun antibiotic in the hospital. It's the drug of last resort we use when nothing else works, like for MRSA.
Right, the heavy hitter. The mechanism of this resistance in VRE is absolutely fascinating and honestly terrifying. We really need to unpack this carefully. The text mentions a specific molecular sequence, D-Ali-D-Ella. Can you walk us through this molecular lock and key concept?
Absolutely. Let's visualize how a bacteria builds its outer cell wall. The wall is made of long polymer building blocks called peptidoglycan. But just stacking blocks isn't enough. To make the wall strong enough to prevent the bacteria from popping under its own internal pressure, these blocks have to be heavily cross-linked together. Now, the end of each unlinked block has a very specific chemical tail sticking out. Two amino acids called D-alanine attached to another D-alanine.
Okay, so D-Ella, that's the structural tail.
Right. And that tail is the exact target for vancomycin. Vancomycin is a massive, bulky molecule. It works by floating in and physically sitting directly on top of that D-Ella tail. It acts like a giant cap. Because the huge antibiotic molecule is sitting there, the bacterial enzyme that comes along to build the crosslink physically can't get to the tail. It's blocked.
So vancomycin just parks a bulldozer in the construction site so the builders can't work.
Exactly. The wall stays loose, weak, and unlinked, and the bacteria eventually bursts and dies. It's highly effective. Now, VRE, the resistant mutant, has acquired a special set of genes called the VANAD gene cluster. This gene cluster acts like a completely new retooling factory for the cell wall. It changes the raw ingredients. Instead of making the wall end in D-alanine, the new factory makes it end in D-alanine and then attaches a molecule of lactate instead.
D-Ella-D-Lac. It swapped out one single amino acid for a lactate molecule at the very end of the chain.
That one tiny substitution changes everything. Vancomycin is a highly specific, rigid molecule. It evolved perfectly to bind tightly to D-Ella via 5 hydrogen bonds. But with that lactate substitution, it loses a critical hydrogen bond and experiences steric hindrance. It literally cannot bind tightly to D-Ella. Its binding affinity drops by a factor of 1,000. It just falls off.
So the bacteria subtly changed the shape of the lock so the antibiotic key doesn't fit anymore.
It changed the lock. But here's the truly genius, terrifying part. The bacteria's normal wall-building enzymes can't cross-link D-Ella either. So the VANAD cluster doesn't just change the lock. It also synthesizes completely new wall-building enzymes that are capable of using the new D-Ella tails. So the bacteria builds its strong wall just fine, the antibiotic floats by completely uselessly, and the deadly infection continues unabated.
That is incredibly sophisticated. And the text says this system is inducible. It requires sensing the antibiotic first.
Yes. It's not running all the time because making this alternate wall takes a lot of cellular energy. The system has a built-in sensor protein on the surface called VAN-S that physically detects the presence of vancomycin in the environment. When it senses the drug, it sends a signal to a regulator protein VAN-R inside the cell, which then turns on the factory to produce the resistance genes only when the antibiotic is actively attacking. It is ruthlessly efficient.
And these genes, this whole VANA factory cluster, they're located on a transposon.
A transposon, yes, a jumping gene. It's a mobile, independent piece of DNA that can easily cut itself out of one bacteria's genome and paste itself into a completely different bacteria. And it transfers easily via plasmids. And it's not just jumping to other Enterococci. The ultimate nightmare scenario, which we have started seeing in rare cases, is that VRE transfers this entire vancomycin resistance factory directly to Staphylococcus aureus,
creating a vancomycin-resistant staph VRSA.
Exactly. Staph aureus is far more aggressive and virulent than Enterococcus. Giving it the ultimate shield against our best drug is a public health crisis waiting to happen. That is exactly why hospitals are so incredibly strict about isolating VRE patients with specialized gowns and gloves. They aren't just trying to stop the spread of a bug, they are trying to stop the physical spread of those specific genetic blueprints before they jump to something worse.
Wow. Okay, just a quick mention of segment 9, the obscure cousins, before we wrap this massive topic up. Nutritionally variant Streptococci.
These are a quirky group. They used to be called Abiotrophia. They're basically picky eaters or defective strep. They cannot manufacture their own vitamin B6, which is paradoxal. If you swab them onto a normal blood agar plate, they simply will not grow. They remain invisible to the lab tech.
So how do you even diagnose them if they don't grow?
You might only spot them accidentally if they happen to be growing right next to a colony of Staph aureus or another bacteria on the plate. We call it satelliting. They physically cluster around the other bacteria, feeding off the trace vitamins the staph naturally leaks into the agar. It's a microscopic, symbiotic relationship right there on the petri dish. They are a rare but very tricky cause of endocarditis because blood cultures often come back falsely negative since the bug won't grow in standard broth without added B6.
That is wild.
Yeah.
And finally, Leuconostoc.
Leuconostoc and Pediococcus. You will usually only encounter these outside the hospital in environmental settings or food production. They are the microbes responsible for fermenting cabbage into sauerkraut, usually totally harmless to humans, but
There's always a but.
they are intrinsically naturally resistant to vancomycin. They just naturally manufacture cell walls ending in DLA-DLAC without needing a special resistance gene. So clinically, if you have a patient with what looks like a strep or enterococcus infection and it is completely ignoring vancomycin, you have to ask the lab to check carefully. Are we dealing with a deadly hospital-acquired VRE, or did this immunocompromised patient somehow just get a bloodstream infection from a harmless sauerkraut microbe?
The things you learn when you dive deep into microbiology. It's a wide, weird, interconnected world.
It really is. It never stops surprising me.
So we've covered an immense amount of ground today. We've gone from the rapidly spreading flesh-eating aggressiveness of group A strep to the stealthy, slow-burn heart valve destruction of the viridans group, the sheer lung-filling inflammatory power of the pneumococcus, and finally, the impenetrable antibiotic-defying toughness of Enterococcus.
It really highlights the stunning evolutionary diversity of this single bacterial family. They have adapted specialized tools to exploit almost every single niche of the human body.
What's the big takeaway for you here? When you step back and look at this entire chapter, what really stands out in your mind?
For me, it's the incredibly razor-thin line between what we consider a friend and what becomes a deadly enemy. We carry pneumococcus in our nasal passages all winter. We carry millions of viridans on our teeth right now. We harbor billions of enterococcus deep in our gut. We are essentially walking biological zoos. And 99.9% of the time, we coexist peacefully. They are a functional part of us.
But all it takes is one tiny slip-up in that balance.
Exactly. A bad viral cold that strips a mucus layer, eating a highly sugary diet that changes the mouth pH, a small cut in the gum from a toothbrush, or a medically suppressed immune system. And suddenly, those quiet neighbors become ruthless killers. They already possess all the tools, the thick capsules, the spreading toxins, the resistance genes. They don't have to invent them on the fly. They are just patiently waiting for the door to be left unlocked.
It makes you appreciate the invisible work your immune system is doing a whole lot more. It is actively holding back a massive, highly armed tide every single minute of every single day.
It absolutely is. And it also highlights the importance of very basic hygiene. Simply wash your hands. Semmelweis proved it 2 centuries ago, and it's still our best defense.
Wash your hands indeed. And after learning about S mutants, maybe brush your teeth a little more thoroughly tonight before bed. Physically disrupt those sticky dextrin fortresses.
I highly recommend it. Floss carefully, though.
Good point. Keep the viridans out of the bloodstream. Well, I'm going to go brush my teeth right now. This has been a deeply fascinating, if slightly terrifying, deep dive into the hidden microscopic wars of Streptococci and Enterococci. Thanks for taking the time to unpack all of this heavy material with us today.
It was entirely my pleasure. Thanks for having me.
And to everyone listening out there, keep exploring, wash your hands, stay curious, and we'll see you on the next deep dive.